Thank you very much for the nice introduction. I also want to thank the organizers to give me this opportunity to give a talk here. I'm so honored and very grateful. So in China, the manned space exploration was initiated in 1992, decades later than Russia, America, and other European countries. But we made some substantial progresses step by step. Next step is going to build up our space station. So you will know that, just as mentioned by our distinguished speakers in this morning, that the radiation is a main risk to the health of our astronauts traveling to Moon, to Mars, the long-term exploration missions. And this space station is a laboratory in the outer space, which provides a unique platform for us to conduct the space radiation research. Certainly the radiation level on this orbit is not as high as the Moon or Mars, but it is enough for us to testify our hypothesis or our findings on the ground. There will be the core module will be launched next year, and there will be 2 experimental modules. And certainly there are several in-module cabinets. For the space radiation research, there are 3 racks we can use. One is called Life Ecological Experimental Racks. This rack is suitable for the studies with plants, small mammals, microorganisms, C. elegans, Drosophila, and zebrafish, and so on. Certainly there will be radiation monitors and life support system, certainly some video or microscopy systems. The second one is called the Biotechnology Experimental Rack. This one is specifically for the studies with in vitro culture system cells. Certainly it's also useful for the protein crystallization, and also some other studies like the nucleic and the protein coordination, biomechanical studies. The third one, sorry I don't have a picture for you, but it is an actual vehicle exposure platform. That one is especially for radiation research. We can use that rack for the studies with C. elegans, microorganisms, macromolecules, but not for the in vitro culture cells. So there are also some other facilities were needed for the biological studies, like glove box, or freezers. Certainly these apparatus are very general. If you need any specific apparatus, you are very welcome to get in touch with us. We can develop one with you and for you. So in 2017, our government officially announced that this space station is not only for the Chinese scientists, it's also open for the international corporations. And so far, actually it's last year in Vienna, the Chinese Space Agency already announced the first series of projects. 70 projects selected from more than 300 applications, among which 50 is from China and 20 from ESA. So any one of you desires to use this station to carry out experiments, you can submit your proposals. There is a website. It's very easy to get in access. And for this biological studies on this space station, Actually, the topic is pretty broad, including the biological effects, non-target effects, risk assessment, radiation detection, biosensors, and crop breeding. So actually, so far in China, not so many institutions are focusing on the space radiation research. But anyway, there are two institutes, as I know, because I founded these two institutes. One is called the Gansu K Laboratory of Space Radiation Research. The other is Institute of Space Life Sciences. You are very welcome to cooperate with us. So we already made some progress on the biological studies, including biological effects, mechanisms, and countermeasures. So I will give you some examples. So first one is the cancer risk. Just as mentioned by Alexander, that cancer is one of the health risks we already know. Just as published in Science this year, the twin study results observed some increased in chromosome aberration, which is related to the cancer risk. Actually, in 2003, NASA already released the epidemiological study that the tumor incidence is about 4.5%, so which is relatively high than the comparison cohort. So in our group, we're focusing on this carcinogenesis of space particle irradiation. So we simulated the low dose rate, high altitude irradiation with human epithelial cells. So we expose these alpha particles, expose our non-epithelial cells to alpha particles. The dose rate is pretty low, it's 6.7 milligram per day. And then we examined the tumorigenesis. We found that the incidence of this tumor, we call it nodule. Actually, sometimes for the control cells, they can also form this module, but not tumor, actually. Actually, the alpha radiation can induce the tumor in a dose-dependent manner. So you can see here, this is a multiple exposure and the long-term exposure. The longest one is 75 days. And you can see the incidence is a dose-dependent increase. But there is no difference between a single exposure and multiple exposure. Here you can see the difference between the single exposure and the multiple exposure in the size of the tumor. So far we have these cells and also the primary tumors in hand. Those samples are at different stages of the lung adenocarcinoma. So we are using these models to try to identify some biomarkers for the lung cancer. And for the mechanisms for the carcinogenesis, we focus on the long non-coding RNA. We identified one long non-coding RNA called CRP3, which is very inducible by the ionizing radiation, especially by the heavy particles. So last year, we published a paper on cancer research. We found that this link RNA can directly bind to G-actin to inhibit the assembly of microfilament, so block the cytokinesis, so that the cells will become binucleated. And we also found this link RNA can interact with LDHA to enhance the glucolysis. It also can interact with BAP3 to interfere the function of the APCC complex and cause the incorrect separation of the sister chromatids So the cells become aneuploidy, and certainly this is kind of risk of the tumorigenesis. And we also confirmed this link RNA can promote the tumor initiation and development. So you can see one piece of small link RNA can play a very important role in the cancer generation. The second one is a non-target effect. So what's a non-target effect? For example, I give you a punch, your girlfriend or your boyfriend will feel the pain. So you can see here, published in PNAS 2008 by a research group from Columbia University. You found that they exposed the stomach of the mice, not the mice, the mini pig, to alpha particles, and then you can find the brain tumor. In our group, what we are interested in is when we give you a punch, why only your girlfriend or boyfriend feel the pain? Not all of the audience in the room can feel the pain. We actually found another mediator. It's called the microRNA 663, which targets the TGFbeta. TGFbeta is a well-known mediator of bystander effects. So you can see here in the directly irradiated cells, its expression is suppressed. Then the TGF-beta can express and secrete it to mediate the bystander signals to other cells. But for the bystander cells, once they got the signal of the TGF-beta signal, the expression of MIR663 will increase to inhibit the expression of TGF-beta in the bystander cells. So that the consequence of this single transduction is that the bystander signals cannot be transducted all over this room. Third one is the countermeasure. You will know that the energy of space radiation is pretty high. So the countermeasure is pretty hard. Even the physical shielding or the medical medicine cannot 100% to protect our astronauts. So in the movie, I think you have some kind of the vision that for the long travel in the outer space, astronauts always stay in this hibernation chamber. So an interesting thing is that we found this no metabolism status of the cells actually is a kind of radio resistant. We found that even for the very high RET radiation, there is a low metabolic status can increase this radio resistance to radiation. And the reason is that the RAC2 subunit of NAPDH oxidase plays a very important role. So you can see here the expression of RAC2 is pretty low in this low metabolic status. So the NADPH cannot be oxidized, and the yield of reactive oxygen species is not so high, so the cells become more radio-resistant. In this case, once we put the cells or our models in a very low temperature or low consumption of the food, so the cells or mammals will become more radio-resistant. This is an easy way to protect our astronauts, I think. But you may ask, what will happen if we ask a lot to conduct some missions outside of the cabinet? Actually, we found another very normal strategy. I cannot show you here today, but maybe in the next meeting, please pay more attention to our publication. A brief summary that we found that low dose or no dose rate alpha particles are tumorigenic in a dose-dependent manner, and ionizing radiation inducible link-RNA, CRP3, is an initiator and enhancer of non-carcinoma. MicroRNA and 663 TGFbeta feedback loop limits the transduction of bystander signals. No metabolism might be used as a novel strategy against the space radiation. So here is our group of members. Dr. Pei did most of the mechanistic studies, and Dr. Hu and Dr. Pei Weiwei did the carcinogenesis, and my former colleague, former graduate student Li Pengfei found this link-RNA. I also want to thank Dr. He from Columbia University and our collaborators at the National Institute of Radiological Sciences, Chiba, Japan, including Dr. Furusawa, Cornish, Hinayama, and Dr. Liu. And let me end my presentation with advertisement. We are going to host the 9th International Symposium on Space Radiation Research and Particle Therapy on May 15th to 18th next year in Soochow, China. Soochow is very close to Shanghai. If you fly to Shanghai International Airport, we'll pick you up there. Soochow is one of the most beautiful and one of the richest area in China. So we are pretty sure you will not only enjoy this scientific communication, but also enjoy the history and the culture there. So look forward to welcoming you in Soochow. Thank you all.
Thank you very much for the presentation. We do have one question by app, but as I read that, if people want to ask at the microphone, please, why don't you go ahead first, then, yeah.
Yes, I had a question about the hibernation. Because when I look at myself, if I sleep more than 12 hours, it's really difficult to wake up in the morning. It's hurting everywhere, and it's difficult. Do you think it will ever be possible to be asleep during more than a week, or maybe more than a week, yes, but more than a month, or the time we see usually in the movies?
Yeah, it's a very interesting question. Actually, we thought about it. For those small animals who has this hibernate habit, they are actually, we did the experiment. They are really radio-resistant during their sleeping. But for human beings, I don't think, I totally agree with you. It's not that easy to totally recover. It takes time. But I think we certainly have to have more things to do, to think about it. But I don't know. But anyway, we'll try.
Thanks. So I'll read the question we have. Oh, that used to be on the screen. Oh, it's there in a different order. So that's quite a specific one about the cell cycle, and it asks whether there are radiosensitivity differences between the synthesis phase, and whether that's higher than in the G1 and G2 phase.
Yeah, it's a very good question, a very scientific question. Actually, for the G1, G0 phase, we use these two cell model because they have the same, very similar or very exactly the same quantity of genetic material, but at different stages, different philosophy status. Certainly, we checked the synthesis. Actually, for the G1 phase, for those cells in the circulating cycle, they certainly have some synthesis going on. So, but for the G0 real cells is much lower. I don't think it's easy to explain why the G1 cells, which is going on this synthesis but relatively sensitive, I think it's not big. Might be because of not DNA repair system is activated during this synthesis. I'm not pretty sure, but that's my guess.
Thank you. We've just got a couple more questions. We'll take at least one of them. Could you comment further on how your biological results, your observations, might inform radiation protection?
Sorry, I didn't.
The results that you've found, how might they be used practically to protect astronauts?
Yeah, actually very similar to the first question. Actually, we, in ancient country like China or India, we have some kind of practice, or I don't know how to name it. Actually, they can calm down people to stay a very low metabolism system without just drink a little water, but no food taking, no energy taking. You can stay at that status for several days, even weeks. So we have this cooperation with some group and some company and try to test this kind of practice or this activity can be more radio resistant. Yeah. I don't know if I answered it or not.
Thank you very much. If there's time for one more question from the mic, if someone goes up immediately. Otherwise, thank you very much to our speaker for an excellent presentation. Thank you.
Thank you.